Hello to our friends joining us via recording. Today we're meeting in the penguin classroom because Dr. Allis has a things for penguins. During today's session, we're not actually going to study the anatomy of penguins, but we are going to review the stuff that relates to unit number 4. Here's a friendly reminder that the unit number 4 exam opens tomorrow. That's Thursday, the middle of the night, so just after midnight tonight. It's open through Friday night at the end of the day, so make sure you're planning a time for yourself to go through and take exam number 4 about the spinal cord and about the special senses. To get us ready for our review session, we work together with our groups, covering a lot of the different topics from the lessons 12 and 13. So the first thing that we did with our groups is we had a couple of statements that were incorrect. And we worked with groups to figure out the ways that we could fix these statements. So let's look at the first statement together here. Our first statement says that if we damage the ventral horn neurons, so my neurons that live in the ventral horn, that would impact our heart rate. Now, there are two potential things that we could fix in this statement. If I want to know who I'm going to damage to change my heart rate, how would I change this word here? It's not the ventral ones if my heart rate changes. Which ones would I blame if my heart rate had some issues? Yeah, exactly. So the neurons that live in the lateral horn neurons, if I damage those, remember that those were the ones that controlled cardiac muscle, which is heart rate. Those are also the ones that control smooth muscle. That's things like digestion or how big your airways are, those kind of things. So the lateral horn has neurons that control smooth muscle and cardiac muscle. What if I do actually damage my ventral horn neurons? What are some of the things that could happen if I actually did damage these ones? What did they control? Ventral horn neurons. Yeah, exactly. So Kira mentioned the skeletal muscles. Yeah, these are what we call our somatic motor neurons. Here, I'll put that somewhere else here. Ventral neurons are what we call the somatic motor neurons. Somatic motor neurons means they control skeletal muscle. They control skeletal muscle. So if you lose the ability to move a part of your body, that's probably an issue with those ventral horn neurons. If we have problems with our cardiac muscle or our smooth muscle, that's probably a problem with my lateral horn neurons. So this statement was a review for us of the functions of the neurons that live in different places. There is also one more horn that we didn't talk about in the statement here, and that is the dorsal horn. Can you remind me, what's the job of the neurons that are in the dorsal horn? Or what kind of neurons are those in the dorsal horn? Yeah, so those are the type of neurons that we call interneurons, and their job is processing and predicting. So the one other place in the spinal cord that we could have damage would be to those neurons that live in the dorsal horn. And if we damage those, then we're going to have some issues with some of those processing and predicting type functions. Interneurons in the dorsal horn. All right, let's move to our next statement. Our next statement asks about shingles. Now, when we talk about shingles, even before we go into what's wrong with the statement here, what are some of the things that we remember about shingles? Fanshin asked a follow-up question, by the way, if we had issues with the dorsal horn. She asked if that could also lead to some motor issues. It could, but we would say that those motor issues are secondary. So on the exam, if I'm asking you about a motor problem, unless I tell you that we predict the wrong motor function, assume that motor stuff has to do with the lateral horn and the ventral horn. I will make sure to use a word like processing or predicting or figuring out when I'm talking about the dorsal horn. Yeah, good question. All right, so let me use my chat here to put some information for us about shingles. So we're remembering that shingles is caused by the virus called varicella. That's the chickenpox virus, right? So when you get varicella as a child, or it's possible to get it as an adult, you get this skin rash that's all over your body. When your body figures out how to treat that rash, when that rash kind of burns itself out, it decides to go dormant. So dormant means it's hiding, it's not replicating, it's just going to chill out with you. And the place that it chills out is something called the dorsal root ganglion. The dorsal root ganglion. And this is where the virus is dormant. So you get the chickenpox, and once you're done with chickenpox, the varicella zoster virus goes and hides out in the dorsal root ganglion. As you get older, as your immune system becomes suppressed, this virus can become reactivated. So that word reactivated means that it starts to replicate again. When it replicates again, it's replicating in the kinds of neurons that we find their cell bodies in the dorsal root ganglion. Yeah, exactly. So my neurons that live in the dorsal root ganglion, this is where those sensory neurons live. So sensory neurons live in the dorsal root ganglion. My varicella zoster virus will replicate, or it will divide and make new copies of itself inside your sensory neurons. Then those copies travel down the dendrites of this neuron. They're going away from the dorsal root ganglion out to the skin. Remember that they affect one dermatome. So where we see the rash with shingles is across one dermatome. When a person is standing up, here comes my really sad drawing of a person. When we've got a person here and we're talking about their dermatomes, remember that when we're on the torso, those dermatomes look like they're going horizontally, so they're going straight across the body. When we're talking about the dermatomes on the limbs, they kind of look like they're going vertically. They go down those limbs. So a dermatome is the area of skin that your sensory nerve, so the spinal nerve, collects sensory information from. So we only see it across one dermatome because we only have one spinal nerve that is collecting sensory information from those places. So when we talk about shingles, shingles is actually not an issue with anybody in the spinal cord, right? Shingles has to do with that thing on the outside of the spinal cord, the dorsal root ganglion. So we would change this. Instead of the lateral horn of the spinal cord, we would say that shingles is a problem with the dorsal root ganglion. Or we could also mention, I'll add to that, I'm going to add and dermatomes. Those dermatomes are the other thing we might say that we can see it, because that's where those rashes come across. Thumbs up, thumbs down. What do we think about this? By the way, Fanshin makes a good comment that it's totally plausible that AMP finals could reactivate your shingles virus. So stress, not good for the immune system, can lead you to have that virus reactivate and give you some trouble. All right, let's move on to our next spinal cord related thing. We are talking about a tract. Now, when I was bouncing between my groups, one of my groups made a really good connection here. Your first question asked you about whether it's gray matter or white matter when we're talking about this particular tract. But my group pointed out that it doesn't matter what your answer to any of these other questions are, whenever we talk about tracts, are my tracts in gray matter or white matter? Where are my tracts always found? Yeah, exactly. My tracts are always found in white matter. They're always in the white matter. When we're talking about gray matter, this is going to be the location of those reflexes. So when we talked about the patellar reflex, when we talked about the flexor withdrawal reflex, so reflexes are what we find in the gray matter of the spinal cord. Tracts are only ever found in the white matter of the spinal cord. So that's good news for you when you're taking the exam. If I ask you where we'd find a tract, it doesn't matter what crazy name is on that tract. It's a tract. So I always know that it's going to be in the white matter. White matter is always where I find my tracts. Now, this particular tract is called the spinoparietal tract. And when we were talking about these tracts, remember we mentioned that their name tells us where it starts and where it's going, or where it ends. So this particular tract has spino as its first name and parietal as its last name. Based on its first name, spino, where might we say this tract begins? Based on that spino word. Yeah, exactly. It looks like spine, right? So this is going to be something that starts in the spinal cord with those spinal nerves, and it ends where? Based on its name. Yeah, exactly. This one's going to be ending at the parietal lobe, which is found in the brain, in the cerebrum. We started in the spinal cord because of spino. We ended in the parietal lobe because of parietal. Based on where we started and where we ended up, would we say this is an ascending tract or a descending tract? Based on where it started and where it went. Yeah, exactly. This is going to be an example of an ascending tract. It's like that thing that was on Facebook lately, right? Like how it started, how it's going. How it started, spinal cord. How it's going, parietal lobe. That's the way that these names of these tracts are. They're like those Facebook posts. I'm just going to say, when I look back at pictures of me and my husband from where it started or how it started compared to now, we are very different people. For anyone who's been with a significant other for a while, you change a lot, right? So how it started for us and how it's going, very different, including but not limited to 2 small children now. So 2 small children and a cat, actually. Let's be fair. We can't ignore Addie because she would be angry at me and would nip at my heels. So 2 small children and a cat. That's how it's going. That's the second part of our name here. This is an ascending tract. When we talk about ascending tracts, are those sensory or motor? Ascending tracts, sensory or motor? Yeah, exactly. Ascending tracts are always sensory tracts. When information goes up, it's always sensory information, which means the corollary to that is that if information's going down, it's always motor information. So memorize those things in pairs because they go together. Ascending is sensory, descending is motor. If we are a typical ascending tract or a typical sensory tract, how many neurons do we typically find in these kinds of tracts? Yeah, exactly. We typically find 3. Remember, in our ascending tracts, our sensory tracts, we've got the first order neuron, and the first order neuron is the one that detects things. So this is the one, for example, that goes out into your fingers to detect stimuli. Yeah, exactly. Teo mentions this is where we have the receptor. Absolutely. First order neuron. Second order neuron is the one that we find it starts in the spinal cord. So it's going to start in the dorsal horn. Where does that second order neuron end up? Where does it go? Yeah, exactly. It ends in the thalamus. The thalamus. Remember from our brain chapter that the thalamus is the sorting center of the brain. So it's going to send that information where it needs to go. It's going to synapse with the correct third order neuron. And that's the one, in this particular case, we're talking about that being the one that's in the parietal lobe. A lot of our third order neurons go to that sensory homunculus. Remove that word. Sensory homunculus. That's where that third order neuron is found. Wink, wink, nudge, nudge. Reminder. Ascending tracts are the only kind of tracts that have 3 neurons. Motor tracts, descending tracts, they only have 2 neurons. So I think maybe we did this before. I can't remember if we did or not. If we didn't make a table where we put sensory tracts and motor tracts next door to each other, if we did not do that, you should do that for yourself. So comparing things like how many neurons we find in those kinds of tracts, what the names of the neurons are, what the neurons are doing or where they're going, those kinds of things. Again, I can't remember if we made a table like that, but if we didn't, you should definitely plan to do that, to compare and contrast. How do we feel about our tract question here? The tracts of the spinal cord. Okay. Well, let's move into special senses then. Everyone's favorite, in air quotes, thing for us to do with special senses is to consider how the process of hearing occurs. So when we're thinking about the process of hearing, hearing starts at the auricle. It starts at the part that you can see on the outside of your ear. So sound waves are collected by the auricle. It's acting like a funnel. It funnels those sound waves into a part of the ear called the external acoustic meatus. Remember from lab, we called this the outside hearing hole, the external acoustic meatus. Now, at the end of the external acoustic meatus, they bump into something that's going to help them get into the middle ear. What's the structure that divides the external ear from the middle ear? Yeah, exactly. That was a mean thing for me to make you type, right? The tympanic membrane. The tympanic membrane. This has tympanic in it, which is like a timpani, a drum. This, in normal person words, is your eardrum. So the eardrum divides up the external ear from the middle ear. There are 3 little bones called ossicles that are right next to the tympanic membrane. So the first one is a bone called the malleus, the mallet, that hits the eardrum. Then we've got the middle one called the incus, and our last one called the stapes, the one that looks kind of like a stirrup. So when my eardrum gets bouncing, each of these ossicles gets bouncing, they start vibrating, and when they finish vibrating, so when the stapes gets the vibration, the stapes is going to bump into the structure that divides the middle ear from the internal ear. Yeah, so dividing up the middle ear from the internal ear is a structure called the oval window. The oval window. The oval window is where we get into the internal ear. And remember that the internal ear is the only part of the ear that's filled with fluid. So once we get on the other side of the oval window, now we're filled with fluid. The first place that we go when we enter the internal ear is filled with a fluid called perilymph fluid. And we really don't need to know much about perilymph fluid, kind of other than the fact that it exists. Perilymph fluid does not help us hear. It's a filler fluid, if you will. The fluid that will help us hear, that we'll talk about more in a moment, is endolymph fluid. So I use that fluid to help me hear. But when my sound waves first get into the internal ear, they're going into a place called scala vestibuli. Scala vestibuli. This is the fluid-filled tube. I know I heard at least one of my groups talking about that model we had in lab. This is the top tube, the first place where sound waves go, into scala vestibuli. And then remember, they bounce and bounce and bounce down scala vestibuli until they find the specific place on the middle tube called the cochlear duct. Cochlear duct. When they find the place in the cochlear duct where they could be heard, they go from scala vestibuli into the endolymph fluid in the cochlear duct. At the very bottom of our middle tube, remember from lab that we have that structure called the basilar membrane. So the basilar membrane is the structure at the bottom of the cochlear duct, and this basilar membrane starts to move since the fluid inside of it was moving. That causes a particular type of cells to hear sound waves. What's the name of the cells that bump into our tectorial membrane to help us hear? Yeah, exactly. My cells that help me hear are called hair cells. Hair cells. By the way, a bunch of other special senses also use hair cells. Who else uses hair cells besides hearing? Yep, smell. We've got olfactory hair cells and gustatory for our taste. Yep, and then our equilibriums as well, right? So static equilibrium and dynamic equilibrium. Here's kind of a big picture way to think about those hair cells. Everything except vision, when we're talking about all of our special senses, every special sense except vision uses hair cells. Vision uses two special other kinds of cells. What were those two other kinds of cells that vision use to help you see? Yeah, exactly. The process of vision is done by rods and cones. The process of everything else, so balance, hearing, taste, and smell, all of those use hair cells. Okay, so the hair cells start bouncing because they're on top of the basilar membrane. They bump into the tectorial membrane, you hear a sound. Once we've heard that sound, we're going to transfer that sound down into the Scala Tympani. Scala Tympani. This is that lower tube from the lab models, the tube where sounds go to die for recycling. So the perilymph fluid in Scala Tympani transports those sound waves to get rid of them. Ultimately, my sound waves are going to leave the internal ear because they're going to bounce out of a area called the round window, out of the membrane called the round window. I'm pretty sure the homework assignment has you put these structures in order. Not all of them every time, but several of them in order. So make sure we have an idea of whether these structures are in the external ear, the middle ear, or the internal ear. And is talking about stuff with, especially when we're talking about stuff with the internal ear, make sure that we know the order that those three tubes go into. Because they got some sketchy names, right? Scala vestibuli, Scala tympani, cochlear duct. Just make sure we know what order they go in. Thumbs up, thumbs down. Or questions. Is there anything I can help you with hearing? All right. So let's do then, we're filling in a table. I think I had talked with you about making a table like this when we first talked about the types of equilibrium. Let's fill in a table together then that helps us to remember what's going on with hearing and also what's going on with our two types of equilibrium, our two types of balance. So let's start here where, and I know I put them in a different order than we're used to on purpose. Let's actually start at the bottom, which is where we started when we were talking about hearing. When you haven't heard anything lately, when you're just kind of waiting to hear a sound, how many messages or how many neurotransmitters do your hair cells in the cochlear duct spit out when you're just sitting around waiting to hear something? Yeah, we're spitting out a few, right? A few. Remember, I like to call them kind of slow and steady. Slow and steady. Oops. So we're not spitting out very many. We're spitting out enough for the reticular activating system to stay alert, to be listening something. So we're not spitting out very many. We're waiting to hear a sound. When we hear something, what's going on with those neurotransmitters, with those messages when we hear something? Yeah, I like it. Kira says craziness. Yeah, this is when we spit out a lot or many neurotransmitters. We're going crazy. We're sending all of the messages so that we activate cranial nerve number 8. Yes, Kaylin has her message all in caps. I hear it for sure. Yep. So we're spitting out all the neurotransmitters to hear a sound. When we're sending that sound away, when we're putting it into Scala Tympani to get rid of it, what's going on with the number of neurotransmitters now when that sound is going into Scala Tympani? Yeah, exactly. When I just finished hearing a sound, when I'm recycling those sound waves, I'm going to have no neurotransmitters. I'm not going to spit out hardly any neurotransmitters because I don't want to keep perceiving that sound. Now, once I've waited long enough, once I know that that sound is gone, I'll end up back at my slow and steady state. But I'm going to take some time and not hear anything before I go back to slow and steady, just to make sure my brain knows the sound is done. So when we're studying the process of hearing, we spit out a lot of neurotransmitters when we're actively hearing something. We spit out no neurotransmitters when we just finished hearing something. And we spit out a few neurotransmitters when we're just at normal, when we're not really hearing any sounds. Let's go over to static equilibrium. Remember that static equilibrium was the one relative to gravity. So relative to gravity means there's 3 positions. Your head is upright, your head is bending backwards, your head is bending forwards. So let's start when we're upright, theoretically what your head's doing right now. What's going on with the number of neurotransmitters when your head is upright? How many are we spitting out when you're at normal, if you will? Yeah, exactly. This is another one that we would say slow and steady. We're sending out some neurotransmitters. Is it when my head bends forward or backwards that I send all the messages? Which am I freaking out about? Which way does my head go? Yeah, exactly. When my head bends forward, I spit out a lot of neurotransmitters. When your head starts to go forward, when you're driving, hopefully not driving, right? When you're sitting in class and you're really tired, your head starts to go forward, that causes a bunch of neurotransmitters to be spit out. We register that as, uh-oh, I'm bending forward. When I bend backwards, when my head starts to go back, what's going on with my neurotransmitters at that point? Yeah, so Kira mentioned a few. I'm even going to clarify, I'm going to call it a very few, because when we're doing the slow and steady thing, there's a ping, and then a ping, and then a ping. But when our head goes backwards, it's a longer time between those pings. So we're sending out a very small number of neurotransmitters. I like that, Kira. Kira added Gwen to all my pings. So we got penguins going on. So very few penguins when our head's moving backwards, a whole lot of penguins when our head's moving forwards, and a normal number of penguins when our head is upright. I like that. We can change this from how many messages to how many penguins. Look at that. Way better, right? It'll be way more fun to study this way now, right? We're studying penguins. We're not studying neurotransmitters. Come on. All right. Last one here, we're talking about dynamic equilibrium. When we talk about dynamic equilibrium, this one is all about spinning. So when we talk about spinning, again, it has to do with the normal state, the spinning state, and the I just stopped spinning state. So let's start with the state where we have tons of penguins. Which of these three is when I have a whole bunch of those messages? When am I spitting out all of my pings or my penguins? Yeah, I love it. Kira's talking about spinning penguins. That's perfect, right? This just keeps getting better and better the longer we do this analogy here. So when you're spinning, we've got a lot of messages, a lot of spinning penguins. They're having the time of their life. It's awesome. When we just stop spinning, if you're remembering dynamic equilibrium, remember how that had the little thing called the cupula? Kind of looks like a little fire thing. When I'm spinning, my cupula bends this direction, and that bends open the channels on my hair cells, so we send out a lot of messages. When I stop spinning, remember that the cupula bends the other direction. When it bends the other direction, that's going to cause me to spit out very few or even no neurotransmitters. So very few or no neurotransmitters. Kira asked if we have to know which way we're bending. No, you don't need to know which way the cupula bends. I won't ask you that. We do need to know, like you mentioned, though, that we're going to bend the opposite direction when we first stop spinning. So when we first stop spinning, we're bending the other direction because now the fluid is pushing my cupula to go the other direction. I don't believe that I actually give you the pictures of the cupula, so I don't think I'm going to ask you to interpret the pictures. We will need to know for dynamic equilibrium these three states here, and have a working understanding of why these things happen. So the one we didn't fill in here, and it should be obvious to us at this point, right, is when we're not spinning. That's another example of slow and steady. Slow and steady. So with dynamic equilibrium, I want you to know that when you spin, that's pushing the cupula in the direction that opens up those mechanically gated channels. And I want you to know that when you stop spinning, that moves the cupula in the direction that pushes those channels closed. That's why very few or no neurotransmitters are coming out. When we talk about bending our head forward or backwards, remember it comes back to pushing on those mechanically gated channels. So I push them open when my head's going forward. I push a lot of them closed when my head is going backwards. Otherwise, there's kind of a basic number of neurotransmitters or a basic number of channels that are always being opened when our head is upright. And yeah, and when we're hearing, we start off with pushing a few of those channels closed. Then I hear a sound and that tectorial membrane pushes all the channels open. But when I get rid of that sound, I end up pushing my hair cells in a way that sound completely stops. I hear nothing. So Fanshin had a good clarifying point for us here. I'll put it in a little box for us. The only one that really completely stops, where we have no neurotransmitters spitting out, is in the process of hearing. With static equilibrium and dynamic equilibrium, when we just finished doing something with the spinning, or when our head's going backwards the opposite way, we'll slow our rate of messages down significantly. With hearing, we absolutely cut it off. No neurotransmitters. Questions about these charts? I gotta draw us a penguin because we talked about the penguins. Let me check Kira's question. The guided lesson said no neurotransmitters for dynamic. Okay, so if the guided lesson clarified it as no neurotransmitters for dynamic, then we're going to go with that, with what the guided lesson said. Yeah, so let's go with that. We will box around there and cross off that. Very few. We're going to go with no on that. Yeah. Apologies on that error there. All right, one more thing that we did in our groups is we talked about our two chemical senses. When we talk about a chemical sense, can you remind me in the chat, what does that mean? Or what are some of the characteristics of something that's a chemical sense? How do they work? What's going on with chemical senses? Okay, perfect. The first most basic thing that we got to know about chemical senses, right? So chemical senses detect chemicals. We detect chemicals, absolutely. And those chemicals, since that's what we're detecting in chemical senses, we're using chemically gated ion channels. Chemically gated ion channels. When we think about detecting those chemicals, absolutely, like Kira's mentioning, we have to dissolve the chemicals in something. I'm gonna put generically there. We've got to dissolve the chemicals in something for us to be able to detect them. So like we mentioned, that's going to be things like saliva when we're talking about taste, or things like mucus when we're talking about smell. You all have hit up in the chat for me too. When we talk about the chemical senses, they use chemicals like odorants and tastants. So odorants are the things that we use in olfaction. That's the chemicals that we smell. Tastants are the things we use in gustation. That's the things that we taste. So let's look at Dr. Ollis's favorite, right? Prediction questions. So we talked about how things normally work. That's what we did in our class together. But I want you to predict what would happen if we had too much saliva or too little mucus. So let's start over here with having too much saliva. What were some of the things we said with our group members might happen if we had too much saliva? What might happen to our sense of taste? Okay, I like it. We got the word dilute in there. Dilute the tastants. I like that. Absolutely. Yeah, so it's going to mess with the sense of taste, right? Is the sense of taste going to get stronger or weaker if we have too much saliva? Yeah, so we're going to have a weaker sense of taste. Weaker sense of taste. Absolutely. I've got too much saliva. Because remember, we talked about this or used a picture that specifically showed mucus and the nose, but the same idea applies here. So I'm going to draw for us, we're going to pretend we're in the olfactory epithelium here, we're in the nose. And remember that on the end of the olfactory epithelium, we had a layer of mucus. So here's our normal mucus layer, or it could also be, I'll put in parentheses, saliva, because these processes work the same. So we've got a normal level of mucus or saliva that's supposed to be there. I've got these little tastants or odorants that are floating around. They come from the environment. They attach to the mucus, and then they have to move through this mucus or saliva to get up to our cells that detect it. If instead of having a nice normal level of mucus or saliva, if instead we had this much, suddenly anything that comes from the environment here has to travel twice as far to get to those cells that detect it. So if I have too much saliva, if we have what's called hypersalivation, which actually, ooh, here's a correlation to make. That's actually something that can happen with sarin gas. It can make your salivary glands freak out and make too much saliva. There are also some medications that can do that too. So if I make too much saliva, or like Kira mentioned, if I make too much mucus when I've got a cold, any of those odorants or tastants have to work twice as hard to get up to the cells that can detect them. And most of the time, it's just not going to happen. So we would have a weaker sense of taste or a weaker sense of smell, if we were talking about olfaction, if we had too much of our dissolving thing. Let's look at the opposite side of the coin, though. What if I don't have too much saliva or mucus? What if I have too little? What if I don't have enough? Do we have any ideas? Kira's voting that the sense would be stronger. So actually, I'm going to blow your minds here. Your sense of smell would also be too weak if we didn't have enough mucus. Any ideas why? Why could having too little mucus make your sense of smell or taste weak? It is the same, Kira. Yep, it's the same if we didn't have enough saliva too. Why? Yeah, so Elapine and Fanchon are kind of on the right track there. Same with Kira. When we talk about a chemical sense, check this out. For us to be able to detect a chemical, we have to dissolve it. That was like our underline highlight star with chemical senses. We have to dissolve them in something to be able to detect them. If I don't have enough mucus, I can't dissolve these chemicals. And if they're just dry and floating around and bumping into my cells can't do anything with a chemical that's not dissolved. So we have to have mucus and we have to have saliva because I need a layer of wet stuff to dissolve these chemicals for my cell to be able to interact with them. If I don't have enough mucus, I can't interact with these things because I can't dissolve them. I can't do anything with them when they're dry. I can only do things with them when I've dissolved them. Best analogy I can give is if you think about all of the sugar that you find in a can of soda versus that sugar that's dissolved in the soda, you probably wouldn't eat all of that sugar straight, right? Like a pile of that sugar. But when it's dissolved in a can of soda, it's much easier, right? A little too easy to have that much sugar. So it's the same thing with our taste and smell stuff. I've got to dissolve it to be able to detect it. Yeah, so corollary, the way Fanchen put it is we have to break down those chemicals to be able to detect them. Absolutely. Yeah, that's what's happening when we dissolve them. We're breaking them down into a piece that's small enough that our cells can detect them. Yeah, so Temiteo mentioned the technical name for this condition, anosmia. Anosmia. I believe that's how you say it, anosmia. That's the inability to smell. And that can be caused by having not enough mucus. That can be caused by having too much mucus. We've got to have the right amount of mucus to be able to smell things, or the right amount of saliva to be able to taste things. So that's kind of the biggest picture with taste and smell. Things have to be dissolved. Don't dilute them too much. Don't not dissolve them. If we do either of those things, we're not going to be able to do that chemical sense. So this would also lead to a weaker sense of smell, a weaker sense of smell. Interestingly, I don't remember if we talked about this together or not, your sense of taste is way weaker than your sense of smell. I think if you search way back in the archives, so toward the very beginning of current events in anatomy, I think there's a couple of articles in there that talk about they tested putting odorants on a spoon to change the way that you tasted your food. So they had like 4 or 5 different flavors that they tried, or scents that they tried, scents that they tried. So you've got a fork that smells like barbecue fire smoke scent, and you put that with a piece of meat, and you taste like it's barbecue meat. Or I think they did it with with wasabi flavor, and then maybe like a fruity flavor. So if you're interested in seeing how your sense of smell affects your sense of taste, there is an article about that in current events. Yeah, so Fanchen said she's curious how they work together. To some extent, they're actually kind of detecting the same chemicals. It's just some of those chemicals end up going to your taste buds, and some of them end up going to your nose. So you actually integrate the information of what you're tasting with what you're smelling to have the overall taste experience. So it's a combination of what you taste and what you smell. Your brain puts them together to tell you what it's like to eat. I always like to joke, by the way, too. If you have a stuffy nose, you've got too much snot, and you can't taste anything, which is incredibly sad when you have that happen, right? You can actually have like half a second or a second of taste when you first blow your nose. Because if you get rid of all that extra snot for just a moment, there's the perfect amount of saliva, or excuse me, the perfect amount of mucus before your body, makes too much again. But there's a split second where you can almost half taste for just a little bit. So next time you lose your sense of taste, think of Dr. Ollis and think of my trick where for a brief moment, you can smell and taste things right after you blow your nose. So make the most of that very brief moment to taste something that you really want to taste. Yeah, so Elapina's mentioning the places in the brain that process those sensations. Yeah, they're pretty close to each other, so it'd be easy to send information back and forth. Yeah, so and Kira's mentioning with coronavirus how that messes with the sense of smell. Yeah, so Audrey, do you still have issues with that? I was reading about a chef in New York who is having to relearn everything because, you know, cooking in a restaurant is so much based on what you taste. And she lost her sense of taste, and it has not come back yet, at least not in the same way. So yeah, I'm so sorry to hear that, Audrey. That's terrible. And yeah, Kira has an aunt that's having issues with that. Yeah. Yeah, for that chef, it has been a very big change for her because that's her job. I can't remember. I don't think I pinned an article about this, but if I remember correctly, the reason they think that the smell is so profoundly affected is actually some of those special cells that were in the olfactory epithelium. If you go back to that picture in your packet, there's some cells that live next to the olfactory hair cells that keep them healthy and nourished and that kind of stuff. I believe they actually think that coronavirus replicates inside those cells that are next to the olfactory cells, and the pressure actually ends up killing those neurons. So it takes a while for those neurons to come back, and the synapses that they had with other neurons may not grow back exactly the same, or just may not grow back, period. So I believe that was the latest thing that I read about that. Yeah, so Audrey said when she caught COVID, there was a burning smell. Yeah, so interpreting things differently. Yeah, that's crazy. Well, this is all I've got planned for today. Are there any last minute topics we want to cover before I turn off the recording for the day? I will mention while we're thinking, let me see, yeah, I do still have it. While we are thinking about things, remember that there is a survey. Let me see if it'll let me paste it onto my slide. There's a survey here for my friends watching. You're not going to be able to click that link. You could maybe copy it or just go into our announcements. It says click here. Oh, perfect. Yeah, Kira said she put it in the group me. There's our link once again. Help me know what we want to review on Friday. That will be our first final exam review session. Our second final exam review session will be on Monday. So plan to attend those sessions if you can. That link that is posted either in the Blackboard announcement or you can see it in our chat here, that is a way for you to voice what you'd like to talk about. So please take a moment, fill that out for me, get your voices heard, and we will have great review sessions later this week. Yeah, Audrey asked about what's due. So I did extend the deadline on the lesson number 13 assignments just so that you can keep going in and working on them between now and Friday. I also gave you a little bit more time on the unit number 4 current events, so I made that due Friday as well. So technically, I don't think that there are any assignments due today anymore, but the sooner you can get lesson 13 done and the current events and anatomy, probably the better. All of that is now due Friday, by the end of the day Friday, which is the same day as the exam. And correct, Kaylin, the exam is spinal cord and special senses. That'll be the exam that is lesson or exam number 4. All right, well, I'm going to turn off the recording. I'll stick around for a few minutes. We can definitely chat together. Otherwise, to my friends watching, I hope to see you on Friday or on Monday. Good luck.